Today we are going to start our next topic, which is protein synthesis. In the past, we've already learned a lot about proteins. So while we're going through this, you need to think of the things we've already learned. What are the building blocks of proteins? Why are proteins important for us? What are their functions? Where in the body are proteins found? So what are the different examples of proteins? And where in the cell are proteins made? As you can see here, the definition of protein synthesis is process of building proteins within a cell. Keep in mind, we already know this word synthesis. Synthesis means to make, okay? We're combining simple substances to form complex substances. So protein synthesis can be broken down into two different stages, okay? The first stage is called transcription. The second stage is translation. You do need to remember the order of which happens first. The easiest way to remember this is that both of them start with the prefix trans, but transcription then has a C and translation then has an L. And we know that C comes before L in the alphabet. The process of protein synthesis begins with DNA. DNA contains the directions for making proteins. If you recall back to when we learned the function of the nucleus, we said that the nucleus directs protein synthesis. The reason why is because DNA is stored there. So DNA is safely stored in the nucleus. DNA should not leave the nucleus because if it does, it gets destroyed. So this is the master copy of the directions, so they always need to be safe, stored in the nucleus. This is why we need to have a helper molecule called RNA. So messenger RNA is going to be the name of our helper molecule that will make a copy of DNA during transcription, and it will then be translated into a code that makes sense at the ribosome. So once again, we know that DNA is found stored in the nucleus. So a DNA, we have a long stretch of DNA. The shape of it is a double helix. Certain sections of DNA that code for making proteins are called genes. Okay? For every gene, one protein is produced. This structure, all the genes are found on the larger structure, the chromosome. And finally, all of the chromosomes will be stored inside of the nucleus of the cell. DNA is the master copy of directions for the cell to make proteins. Again, we do not want the DNA leaving the nucleus because it could be destroyed. DNA will safely be stored there. We need a helper molecule involved to make a copy, and as long as DNA isn't destroyed, we can go back and continue to make these proteins. If you think back to when we learned about the different types of nucleic acids, we did mention both DNA and RNA. So here's where RNA is going to come in during protein synthesis. RNA stands for ribonucleic acid. RNA is a copy of DNA that goes out into the cytoplasm to tell the cell what to do. You can always make more RNA, so it's okay if it gets destroyed. And we are going to learn two different types of RNA that will be used with protein synthesis, are mRNA and tRNA. Now, if you look at this diagram over here, you can notice some differences between our DNA molecule and this messenger RNA molecule. Right now, I want you to jot down a few differences that you notice just by looking at this diagram. This chart compares the differences between RNA and DNA. Hopefully, you are able to come up with some of these. mRNA or RNA is going to be single strand. We know DNA is a double helix, so one strand versus two strands. Next, if you look at the building block components, the building blocks of both of them are called nucleotides, but look at the difference here. They both have a phosphate group, they both have a nitrogenous base, but look at the names of the sugar. OSE is our ending for sugars. Look here. For RNA, it's called ribose. But DNA, it is called deoxyribose. So we have a different sugar. The last difference you can see in terms of the nitrogenous bases. For RNA, there is uracil, represented by the letter U. In DNA, we have our thymine, represented by the letter T. So instead of having thymine like we're used to with RNA, it is replaced by uracil. So during the first stage of protein synthesis, called transcription, this is the process where mRNA is made from one gene of DNA. So M will stand for messenger, and the mRNA will take the message from the DNA all the way to the cytoplasm. Specifically, it's going to end up at a ribosome. The different steps for transcription are listed here. Step number 1 would be to unzip one gene in DNA because we want to make one protein. Step 2, we will match up the bases to one side of the DNA to make mRNA. Step 3, once the mRNA is made, it will be detached from the DNA, and finally, step 4, it will go out of the nucleus and into the cytoplasm. This diagram here shows the process of messenger RNA transcribing one gene of DNA. The outer green section right here, this is our DNA molecule, our original DNA. So you can see how it has unzipped from each other. So the two strands once were bonded, now they're unzipped, those hydrogen bonds are broken. Now we have messenger RNA nucleotides being added to the exposed bases. So where there was a C on DNA, we now have this G. Where there was an A, we have a U. Where there's a T, there's an A, G, and a C. Remember, there's no T, so where you would have normally bonded a T to this A, instead you have added a U right here. So, during transcription, in order to figure out the mRNA sequence, again, where there's an A on DNA, you will have a U on RNA. A C would go to a G, a T goes to an A, and a G goes to a C. So if one strand of DNA had the following base sequence, right now try to write down what the mRNA sequence would be once it was transcribed. Okay, hopefully you were able to come up with C, U, C, U, U, G, A, U, C, A, U, G. Here's a diagram that goes through transcription looking at this whole cell. So we start with DNA in the nucleus. It is a double-stranded molecule. Once mRNA makes a copy of this DNA, it will be a single-stranded sequence, and it will leave the nucleus and travel to the cytoplasm. Once we're in the cytoplasm, we need to carry out the next stage of protein synthesis. This is called translation, which is the process of joining amino acids together to make a protein. Recall, the building blocks of proteins are amino acids, so we need to join these simple amino acids together to make the large complex protein. And don't forget, there are 20 different kinds of amino acids. So for the process of translation, step 1, the mRNA will attach to a ribosome. The ribosomes are either going to be free-floating dots in the cytoplasm, or they will be attached to, if you recall, the endoplasmic reticulum, the rough ER. Step 2, we have our other RNA molecule involved here. tRNA, which stands for transfer, is going to bring amino acids to the ribosomes. The amino acids will join together to make a protein, and finally the protein will detach from the ribosome. Right here we can see, if we start up at the nucleus, mRNA has been produced during transcription. So we have our single-stranded mRNA molecule, and it's going to travel to this ribosome. It will be straightened out at the ribosome, and for every 3 bases on mRNA, that is called a codon. Every 3 bases on mRNA is called a codon. And that is an important word that you need to know. The codon is going to match up with every 3 bases on the tRNA molecule called the anticodon. So those are going to be complementary to the bases on the mRNA. And this is how the transfer RNA knows which amino acid corresponds for the codon. The amino acids will be carried by the tRNA molecules, and if you look, the tRNA really look like the shape, the letter T. As you are observing this, note that only one amino acid is brought by each tRNA molecule. The amino acids will be bonded to each other by peptide bonds, and finally you will have a long strand of amino acids. It will be folded into a certain shape, and you will end up with your protein. The last very important part of protein synthesis that you need to know is how to determine which amino acid will be brought based on either the mRNA sequence, the codons, or the tRNA anticodons. This chart right here provides the mRNA codons. So, for instance, if we start with, let's say, GGU. So if GGU is the codon for mRNA, you're going to find the first base on the left-hand side, which would be G. Second base is up top. So our second base is G, right here. And what you're going to do is you're going to go down and over until you meet. You can use this third base on the right. I always just look in the box and I find GGU. So here's my GGU. So the name of the amino acid is glycine. Right now, what you should do is practice. If you have a DNA sequence of AAA, DNA. Right now, take a minute and figure out first what AAA would be transcribed into for the mRNA, and then determine which amino acid it would code for. Okay, hopefully at this point you realize that AAA is going to be transcribed into U. If we find our first base on the left, we have our U. Second base on the top, our U. And this one's easy. You go down and over and you really just end up in this first box, and hopefully you see that phenylalanine is the name of the amino acid that it codes for.